So by that, I want to tell you a little bit about quality assurance and quality controls in metabolomics, which is quite important. As you can see, when you have a big biobank, as we saw before, then it's important to run some kind of quality assurance and quality control systems. So why would we use that? It's ultimately, by the end of the day, you want to have a very nice study, and you want to demonstrate it's of high quality, so it can benefit as many in the community as possible. You don't want to report a low quality study, so it's important to have some good quality control and quality assurance systems in place. And finally, you also want to, at the end of the day, you want to share the data in repositories maybe, and Justin will come back and talk about that later today. And then many times today, publications, journals, you have to report your study in the end. Then you also have to provide some kind of a description of what quality assurance and what quality controls measures you have been using in your study. So, and after my talk, I will introduce this topic to all of you. And then after my talk, Morten Danielsen from MS will come in and show how it works in real life from a company side. So QA and QC is under the umbrella called the quality management system. And there are some quality controls that are happening just before analysis. That could be something about study design descriptions. Is there any sample handling and study sample storage descriptions, sample tracking, and so on. And then during analysis, there's also quality control checks. Normally, before we use our systems, we do system stability checks. We have in studies, we typically have blanks. And that's also in the study you're going to work with the next couple of days. There's some blanks. Maybe there's some descriptions of internal standards in the study also, or you're adding that you want to follow. And then there could be a pooled matrix of QC samples that we will get back to in a few moments. Then there could be after data acquisition, then there could be something about data storage and archiving. It's a very good idea to have that in place and some descriptions of it. How are you going to do the data review? Are there any quality checks? Are there any extra persons that are looking into the data and are getting the same results? Maybe you're using a PCA for checking for outliers. There are some descriptions about how to annotate and identify the metabolites. And there's some acceptance criteria for filtering and so on. And maybe there's also this one from the pooled QC samples. Maybe you review if there's high variation of this QC sample of a certain metabolite. Then maybe you would reject that metabolite for further studies, because maybe it's only present in very few samples, or maybe there's a very high variation between the pooled sample injections. And then you have the quality assurance system, and we looked a little bit more into that, and that's happening before any analysis, where it's standard operation procedures, and up here we cannot see, but it could be monitoring of temperatures in the room, power failure, and so on of the instruments. That's a very good thing to have these logs. So everything is typically entered in the logbook of the instruments, and the instruments are of course following some kind of a maintenance schedule, and how often are we calibrating the mass spec and so on. That should all be within the quality assurance systems. And documentation and audit trails. So if there's any changes to the standard operation procedures, or if any samples are undergoing another procedure than all of the other samples, then you document it in such a thing. And if we then jump into about data storage and repositories, then it would be a good idea to think about, like, when you acquire the data, how are the data going out to the clients or the person that are working on the data. So in our lab, we are using a network-based approach where all the data are being stored on one computer, and then afterwards we are copying it to network drives where there's a mirror of the data quite soon after, and then the client can enter that data. And then after the data analysis, we can also put it in an archive where it can be stored for many years. So if you are involved in Horizon projects, for instance, then you are requested by law that you have to store the data for at least 7 years after the project has ended. So that's typically what we do. We place everything in an archive when the data analysis is finished. And then there's a lot of repositories that Justin will talk about and how to share it with everyone. So the quality control, it's something that we do during or after the analysis. And one of the things was the suitability test. So it will demonstrate if the platform is good enough. And what we typically do in our lab is that we have a standard that has been spiked into a matrix with at least 50 different components, metabolites or pharmaceuticals, and then we run that on the platform, and then we check if the retention times are good enough, the mass accuracy is good enough, and so on, the intensity of the samples, intensity of the metabolites, and if it's good enough, then this platform is okay to use. Then there's something about this QC pool, where it has several purposes. One thing is to conditioning the analytical system. The other thing is to look into the reproducibility of all the injections that you do in the study, and it's also to monitor and correct for systematic errors, so we can normalize our data after this, and I will come back to an example of that. And then we can also filter the variables, all the metabolites and intensities according to this pool QC. And then we could also use it in a dilution study, and maybe some people are already doing that here, where we want to, if we want to compare sample groups, then maybe it's a good idea to know if to demonstrate if you have a metabolite that is half in intensity, then it should also be the half of the concentration. And you will do that in a dilution study where we typically take this pool sample and we dilute it a factor of 2, a factor of 4, and so on. And then we hopefully see some kind of a linear relationship of the signal. Then there's process blanks or procedural blanks or process blinds. It's very important to maybe there's things you introduce during your sample prep or handling of samples, then you can filter out this signal from this sample. And then you have maybe a QC spike sample also, where you fortify the pool sample with some metabolites to confirm the identity of metabolites. And then we have long-term reference materials. If you want to normalize across batches or bridge data sets, then it's a very good idea. And there's an example here where this long-term reference material has been injected every year of all the red injections here. And this is for a single metabolite, you would say. And this is the second batch where it's the same sample that's injected, and the third one. And then you can simply normalize this data and use all of the data in a study. Then finally, I just have an example here of a sample sequence. What we typically do. Typically, we use solvent blanks first on the platform, and then we use this system suitability check, and then we use a solvent blank again and a process blank. And then we start injecting quality controls to conditioning the system, because there could be active sites on the chromatographic columns and so on that needs to be saturated. So we are conditioning the system by that. And after that, we typically run a sample with DDA, as we heard this morning from Daniel and Farina, where we are fragmenting all the metabolites to get data for annotation. And all of the other samples are basically recorded and just in full scan mode with this strategy. And then we go into all the samples. This is in randomized order, of course, and it's kind of balanced. For instance, if this study is about looking into some kind of diseases, and you have like 50% female and 50% men in the study, you want to balance so you don't have overweight of men in this group or females. So you balance it out, so we have 50% of each in these groups. And then after the samples has been injected, then we continue with a quality control and another block of samples and a quality control and another block of samples, quality control again. And this is what we can see here on this plot here. So the blue dots are quality controls, and the yellow dots are samples. And this is the intensity plot for a single metabolite in a study, where it maybe contains thousands of metabolites. And as you can see here, there's a drift of the instrument, a performance of the instrument. And when we do this, then this QC should give the same signal in all injections, but it does not, because the system will go down in performance typically. So we can normalize this later on with the data analysis. And then we finish up with some more identification only injections of the pool QCs, and maybe a process blank and a solvent blank to check carryover. And then a system suitability test again. And this is an example up here of a system suitability test we did in the beginning of the analytical sequence. A very nice and beautiful chromatogram with a lot of peaks, and this is what happened in the end. So this does not look so good. So basically, we had to do something, clean the instrument, change the sample prep to get the same signal again, because you can see here when you compare these two chromatograms, they are completely different, and we had to change sample prep parameters and then restart the sequence again. So any questions about the QA/QC for now? Yeah.
What mass spectrometry platform did you run this on? And is the drift you see here translatable to different platforms?
So this picture here is an example from a paper I referenced down here, and the chromatograms are not associated with this figure here, but the chromatograms are from a nano LC chromatograph. We have hyphenated to an ultra in our lab. So, but we typically see this, that you have a very good signal in the beginning, and then it goes down in performance across, I don't know, 50 injections or something like that. Yeah.
You see a similar effect also in different platforms?
Yeah, it's a typical effect you see, yeah. And then you have to change your strategy a little bit. Maybe you need to do more sample cleanup. Maybe you even have to dilute your samples sometimes. Use less material, yeah.
How do you decide which task to balance in your group. I mean, I get that you try to balance man and woman, but you could also decide to balance for older people, younger people, or students or dads from different regions. But
yeah,
how do you decide?
Well, it depends a little bit on your study design, I would say, and hopefully you know what to study before you just go completely untargeted. But maybe this is a controlled animal study with different treatment groups of a drug that is being tested. Then you have to kind of balance the different groups here in the different brackets here of samples. So don't run like all females or all in the high dose in one end of the study, because maybe you have a situation where you have a very good signal in the beginning and then a low signal in the end, and then you end up drawing the wrong conclusions. Maybe. Yeah.
Randomization already prevented if we have.
Yeah, but it doesn't all the time when you run a random function. Yeah. Yeah. So that's why we use this balancing or bracketing function, and we typically run this pool quality control every 4 to 6, 8 samples or something like that. It's depending on the size of the study. Yeah.
The quality control need to be different from the sample itself, or it needs to be similar?
Yes. The quality control could be any sample, but typically, if you want to do it in a good way, it's typically a pool sample of the study. We will take a little bit of all the samples into a pool. And you could do that on the sample level before extraction, or you could do it after extraction.
5 samples separated within each quality control. Is it arbitrary, or just like I can do it like 10 or 5? What is the?
It depends on your size of your study, but typically it's every 4 to 6 to 8 samples, and you have a quality control. Yeah. NIST, I didn't know NIST had a quality control sample. NIST is typically a library, or is it also a? Yeah. Okay, yeah, a standard reference material. Yes, yeah, you can do that. Yeah. So the NIST standard reference material is typically from a human biological sample. Normally, yeah, we don't use human samples that often, but we may be looking into zebrafish or daphnia, and then we have to make our own material from all the samples. Then we take a little bit of everything and combine into a pool. Yeah. Yeah. All right. Any other questions? Yeah. Yeah. Yes, so I would ask Morten Danielsen to come forward, and then we just switch presentations here.